Epigenetics is the study of heritable changes in gene function that do not involve alterations to the underlying DNA sequence. In simpler terms, it's about changes in gene expression, how genes are turned on or off, and can be passed from one generation of cells to the next. The DNA in our cells is wrapped around proteins called histones to form a structure called chromatin. Epigenetic modifications involve chemical changes to the DNA molecule or the histone proteins, influencing how tightly the DNA is packaged and consequently how easily genes can be turned on or off. One of the key mechanisms in epigenetics is DNA methylation. This involves the addition of a methyl group to a cytosine base in a DNA molecule. When a gene is methylated, it typically results in the repression or silencing of that gene, preventing it from being expressed. Histone modification is another crucial aspect of epigenetics. Histones can be chemically modified by adding or removing certain chemical groups, such as acetyl groups. These modifications can alter the structure of the chromatin, making the DNA either more or less accessible to the cellular machinery responsible for gene expression. Acetylation of histones, for example, often leads to a more open chromatin structure, allowing genes to be expressed. The presence or absence of certain chemicals or environmental factors can influence these epigenetic modifications. For instance, the addition of methyl groups to the DNA can inhibit the binding of transcription factors, making it difficult for the transcriptional machinery to access the gene and initiate gene expression. Reduced methylation can make the gene more accessible and increase the likelihood of gene expression. Adding acetyl groups to histones generally opens up the chromatin structure, promoting gene expression. Removing acetyl groups can lead to a more condensed chromatin structure, inhibiting gene expression. These epigenetic changes can be influenced by a variety of factors, including environmental exposures, lifestyle, and even experiences. The field of epigenetics has profound implications for understanding how genes are regulated and how environmental factors can impact gene expression and potentially the development of various diseases. It's important to note that epigenetic modifications are reversible, which means that they can be dynamic and responsive to changes in the environment. This dynamic nature is one reason why epigenetics is an exciting area of research with implications for fields such as medicine and personalized healthcare. A recent 2023 study, the link to which will be in the description, found that prolonged marijuana use is strongly correlated with increased methylation near the AHRR gene, with a p-value of 0.000, there's 9 zeros there, 5. To make it clear, any p-value below 0.05 is typically considered significant in statistics. Whether or not continuing this research will allow us to quote-unquote shut down certain unfavorable variants in our DNA is unfortunately at this point unknown. Thanks for watching. Goodbye.